Hello, this is Candace Holman, and myself and Miss Barb Eckes are going to provide a presentation on hemochromatosis. So in getting started, hemochromatosis is most often an autosomal recessive genetic condition that results in the excessive absorption and storage of iron in the cells of the body. Now, the CDC estimates that the prevalence of the homozygous form in the United States is approximately 1 in 200 to 1 in 250 people. The disease primarily affects those of Northern European descent or Caucasians. Mutations of the genes that control hepcidin regulation on the HFE gene are responsible for up to about 90% of the diagnosed cases of hereditary hemochromatosis in the Northern European population. Now, hepcidin is a protein produced in the liver that's responsible for iron storage and absorption in the digestive system. Mutations that can be carried by parents and contributed to their offspring can be homozygous, where each parent contributes the same defect on the HFE gene, or when each parent contributes different mutations on the HFE gene on chromosome 6, it results in a compound heterozygous defect being present, which can still result in the phenotype of hemochromatosis. Now, there are 4 main types of hemochromatosis, and types 1 through 3 are inherited as autosomal recessive traits, and all 3 result in a hepcidin deficiency leading to the iron overload. Type 4 is inherited as an autosomal dominant trait and affects a different protein, so I will describe that later. Type 1 is the most common form described on the previous slide, where there's an HFE gene mutation. Typical onset of the disease for these folks is between the ages of 40 and 60 for men, and it usually follows menopause for women. The majority of people who have type 1 hemochromatosis have inherited 2 copies of an HFE gene mutation caused by substitution during encoding called the C282Y mutation on chromosome 6. It's been found that the penetrance of this form of hemochromatosis is between 2 and 28%. So of those who carry the genotype, 2 to 28% of them will show the signs and symptoms of hemochromatosis. Type 2 is a juvenile onset form of the disease, which tends to be more severe, and it is caused by defects in the HJB gene or the HAMP gene. It results, in addition to the other common symptoms, in this form particularly, in decreased or absent sex hormones, and typically in cardiomyopathy or some other form of heart disease, frequently by the age of 30. Type 3 hemochromatosis has its onset during young adulthood, usually by the age of 30. This form is caused by a mutation in the transferrin gene, and hence is called TFR2. It's more common in sort of the southern European places like Japan, Brazil, and Italy, but nonetheless is still relatively rare compared to type 1, for example. Type 4 has its onset between the ages of 40 and 60 and presents very similar to the way type 1 presents, but is inherited in an autosomal dominant fashion. Type 4 is caused by an SLC40A1 gene mutation, and this form is also called ferroportin disease because the mutation causes a disruption in the ferroportin proteins involved in both iron absorption and the movement of it throughout the body. So what are the signs and symptoms of hemochromatosis? And another indication that may be seen is this sort of to summarize the genotyping, most people with hemochromatosis will be homozygous for a C2A2Y defect of the HFE gene. This mutation is the result of a guanine being substituted for an adenine, which results in tyrosine rather than cysteine, or a completely different amino acid. Some people with hemochromatosis will be heterozygotes with perhaps a defect called H63D and the C2A2Y mutation. And those folks can also present with the phenotype of hemochromatosis. In the case of type 4 hemochromatosis, if just one parent contributes a defect in the SLC40A1 gene, the person may have the phenotype of hemochromatosis. With regard to family, when an individual is diagnosed with hemochromatosis, it is recommended that first-degree family members also be offered testing or screening. Then usually they start with lab work for the iron levels. Of course, siblings of affected individuals have a 25% chance of having the disorder. And if the patient has children, testing of the father will provide some insight as to whether and to what extent the children are at risk of hereditary hemochromatosis, and or whether they're simply carriers of the mutation. Initial screening, as I said, is likely to begin with serum ferritin and transferrin saturation levels in those between the ages of 18 and 30. As far as the ethical and legal concerns that might surround a diagnosis of hemochromatosis, there could be some stigmatization or fear of stigmatization by those that don't have a lot of knowledge about hemochromatosis or understand the disease, particularly if one is struggling with, say, cirrhosis of the liver as a result of hemochromatosis. They may be perceived by others as automatically perceived as being an alcoholic and what have you. There's also possibly some stigmatization brought about by the fact that there are a lot of blood banks that will not accept donations from people with hemochromatosis. I do know that one of them is the Red Cross, one of our major sources of donated blood. So that too can feel a bit stigmatizing. Discrimination, there may be some concern about discrimination when it comes to being able to obtain life and disability insurance, and parents who have discovered that they carry the gene mutations may feel some guilt over what they may have passed on to their children. As far as whether there are legal concerns like the duty to warn, I don't think any cases have been brought about, at least not that I could find. This is not an infectious disease, of course. It's a genetically induced disorder. And I do know, though, that there is some consideration being given to adding it to the list of actionable variants, meaning when people have whole genome sequencing done, they would be informed if particularly the C282Y homozygous mutations are present. But so far at this point, or at least as of June of 2017, the American College of Medical Genetics and Genomics has not recommended that it be included.